So this is our presentation on Artemia, and we focused on magnetotaxis and their response to a magnetic field by me, Amanda.
Harrison Freeman.
I'm Matt.
I'm Sajal.
And I'm Savannah.
So when we researched our topic, we found a paper about how when Artemia cysts are exposed to magnets, it can actually increase their rate of hatching. And this wasn't done too long ago, only 10 years ago, so it's up to date. And then another person looked at the response of bacteria to magnetotaxis, and it was positive in that the bacteria gravitated or went towards the magnets. And they also found that a lot of mammals or aquatic mammals and bacteria tend to gravitate towards the geomagnetic fields or lines of the earth, and they use these to find their breeding grounds or follow food, and just as a form of navigation. So salmon are well known for doing that in the Pacific Northwest, and because they go from up to Alaska to, I know, Washington, and they do use their sense of smell as well, but they also depend on magnetotaxis. And other aquatic mammals like, I think, dolphins and some types of whales, they also depend on it to go to breeding grounds and remember where they are. Yeah.
So the question that we were just dying to figure out was, do Artemia respond differently when a magnetic field is present?
And here's our hypothesis and groups. So we were actually very hard pressed to come up with a hypothesis because although we found a lot of background information to that some aquatic animals do respond to magnetotaxis, we also found that there's not a lot of articles about aquatic animals responding to magnetotaxis, and therefore we kind of assumed that there wasn't a lot of biological reason for the response. But the hypothesis that we came up with was that if the Artemia are exposed to a magnetic field, then there will be a higher amount of Artemia because their food and bacteria, as we found in the articles we did find, respond positively to magnetic currents. And we are guessing that the Artemia will follow them, as just everyone needs their food, right? And our groups was the magnet then and no magnet that we were testing. And so then here was our null hypothesis, which we felt the need to put in since we partly expected no response. If the Artemia are exposed to magnetism, it will have no effect on their behavior because that attribute will not benefit their biology, because as I was saying before, we could find very little evidence that they needed magnetism in any way other than possibly in their food.
So based off of our hypothesis, we predicted that there will be a positive magnetotaxis response in Artemia, and more Artemia will be found in the chamber closest to the magnet. We also predicted, based off of our null hypothesis, that there will be no change in the behavioral response of Artemia when a magnet is present, because it does not affect the food supply of the Artemia and it doesn't benefit the Artemia in any way. So to put it in other words, Artemia will show neither a positive or a negative magnetotaxis.
So in order to perform this experiment, we relied on a choice chamber, which is shown on the slide. And basically how we did this was we put one side with a magnet on it, and then the other side was the controlled side because it had nothing else done to it. And basically we used a 36 ppt solution, but the people who were in the lab used the 27 ppt solution, but it did not necessarily have an effect on the Artemia itself, because no matter what, it would have the same solution on both sides. And so the way we performed this experiment was we inserted the Artemia in the center of the choice chamber. And then after that, we let them kind of decide which way they wanted to go. And we let them sit for 10 minutes to just see how much it would change. And then after that, we used a syringe and kind of took out all of the Artemia. But in order to prevent any further movement between each side, we inserted clay into the middle so that there was no chance of them switching while we were trying to extract it at the same time. And then after that, we also relied on a coffee filter to count how much Artemia was in the side that contained a magnet and the side that contained no magnet. And so then we put it through the coffee filter paper and then used a magnifying glass in order to count how much Artemia was per side.
So here are the bar graphs for our data. On the left, we have our average, which was for our entire data set. And on the right, we have our data for each trial. We did 4 different trials. Unfortunately, for trial number 4, which was the in-lab trial, for like the in-person people actually in the lab, unfortunately for that trial, basically most of the Artemia were dead. So they couldn't really pick a side because they're dead and they can't move. Yeah, so overall we found that, as you can see, there were slightly more Artemia in the no magnet portion of the choice chamber. But as we'll get into in the interpretation of the data, the difference here is not that big. And you can also see throughout each specific trial, the differences were also not that big in between each side. So that led us to believe, as we'll go over in the interpretation, that the results were not really down to magnetotaxis at all. So our interpretation of this data, as I briefly mentioned, there were slightly more Artemia in the chamber with no magnet, but the difference is negligible, really. Our hypothesis is not supported, which was negative magnetotaxis. And so this negative magnetotaxis may show that the Artemia are not attracted to magnetic field, but really we can't really say that confidently because for one reason, the difference is so small, and also because when we did our chi-square calculations, we came up with a p-value that was greater than 0.05, which basically means there's no statistically significant difference in the data, meaning that the data is most likely random and not associated with magnetotaxis. And here are our citations. Thank you for listening to our presentation.
Thank you.